Okie dokie. Wait. Okay, it's actually recording this time. Yay! Okay. I am trying to figure out how to like record these videos in like a productive way that like doesn't that is like candid to exactly how I'm finding the information. And that would be creating a bunch of tabs, and eventually they become quite indistinguishable. But before that can happen, what I would like to do is go through the thought process that is here, and like why a lot of this stuff is interesting. And I think I found something really cool, something really interesting, and it has to do with like an adorable animal, an axolotl. Axolotls are so cute, and they're in Minecraft as well. Hold on, oh wait, I want to see an. I have not seen an axolotl in Minecraft. Actually. Also, there probably will be lots and lots of tangents. Yeah, oh, these guys are absolutely adorable. Oh my god, oh my freaking god. Yeah, this is cinematic. This looks like a LinkedIn profile picture of this guy by the pond, just creating, spawning. Anyway, my name is Cole, and I am a molecular biologist. I do research at the University of North Carolina, and I do epigenetics research. So basically, how your DNA breathes. Because your DNA needs to do a lot of things, because you have the exact same DNA in every single one of your cells. Like all of them. Same stuff. But for some reason, we have different stuff. And that is just because the way that the DNA is accessed in all of those different tissues is very different. And that accessibility is governed by this interaction of... Oh, I'm going to histones. Sorry. It's very easy for me to start talking about histones. But for good reason. Histones. Yeah. What is this stuff? Okay, so if you were to look at a like strand, if your actual DNA like under a microscope, like particularly a scanning electron, like micrograph. Yeah, hold on. That's probably a better start. SEM chromatin. Beads on string. I said swem. LOL. Hopefully you know what that means. Oh yeah, this stuff. Okay, so this is your actual information in your cells. This is the actual stuff. And so it is the double helix, like you know it, just attached always to these little histones, these little beads. These are proteins. So you have a bunch of stuff in your cells that are doing an incredibly important thing. A couple of the like key players are DNA, RNA, and then you have proteins. And these proteins are composed of these sort of, they are like the workers of the cell. They're like these nanomachines that are versatile in their composition and charges and sizes and structures and compatibilities and functions consequentially. Because all it has to do is be made, and because of the way that all the individual components have been stitched together, it will fold into a specific shape, and that specific shape will give it the ability to do a specific thing around the cell. And so for the case of this protein, these histones, let's look at these guys, or yeah, beads on the string, so you got the DNA and you got the histone. Let's see. Yeah, so a closer look at these guys, they look like this. Very, like, generally speaking. It's an octamer, so there's 8 of these little core particles. So these core particles are very stable. They bind together very strongly, and they have lots of amino acid residues. Each one of these is like under 200 amino acid residues. So this is a big thing composed of many, many small parts. And these parts up here, this whole tail, each one of these guys has a tail, and the tail is sort of wibbly-wobbly. It's very labile. It is out there, and we call it disordered. It really doesn't have a predictable location. If we were to try to guess, okay, what's it doing at this given time? Who knows? And yeah, so it's important. But the DNA will wrap tightly around this because DNA is super negatively charged, and histone is very positively charged. And there are certain like access points. Ooh, this is another one. Yeah, looks like this. So this all wraps about 147 base pairs of DNA, and yeah, and there's like a little bit of linker DNA in the middle. And so what are the tails doing? The tails are essentially, I don't know, Axolotls, micro. Histones, nucleosome. Yeah, PTMs, PTM. Okay, so the tails are super important because their amino acid composition allows for them to get modified. These little bubbles here, these are the possibility or possible sites where in real time within your nucleus, these guys can get acetylated, modified, so a little pop that changes the charge. When they make this bond, the tail is now structurally and chemically different. And so, as much as we don't really know what exactly it is, how it is behaving, if we find that there are acetylation, it has a lot of acetylation, we get an idea of the structure around here, because modifications of the tails make the histone a little bit less positive for the DNA that it is associating with. And so when that happens, the DNA gets loose and it becomes accessible, because the physical machinery that is to turn this DNA in the middle here into into anything, and to copy it, to make it into RNA so that it can actually do a job, it has to be physically accessed by the machinery. Let's see. Oh, RNA filter. Paul, what is up, Paul? Yeah, RNA Paul too. Big guy. Like, this is a huge protein relative to Parnatal. Yeah. Okay. Like, the proteins are so complex. Like, this is what they actually look like. Or, well, like, this is, like, how dynamic and, like, turbulent and sort of unique the individual components of the protein can be, and how specialized their functions are. So, like, it could be that this entire thing is to dock on a very specific substrate, and then whatever is up here, pull to CTG. Okay, I'm, like, totally just... Like, okay, I don't actually know what I'm looking at. You have to color code these things for good reason and for them to be, like, decipherable in the slightest. But it could be that this entire structure is to get a small chemical reaction up here that occurs, like, on whatever substrate it's binding to happen. So it needs to dock, and then it, like, brings, like, magnesium or methylation or acetylation, and it places it. And these guys are doing this so often in the cell. In the cell, this is like there is a storm, an orchestra at all times conducting these reactions. And that is how your DNA breeds. Because when the reactions are toggling on and off, and histone is getting acetylated and unacetylated, it's going from closed to open, close, open, close, open. So, yeah. So your DNA, it's really helpful for your DNA to have these proteins, because now it can condense itself, so it's harder to, like, break. It can open in dense areas so that it can repair what is there, and it can make it like the entire thing open enough to be completely replicated. So the versatility of histones correlates to the versatility of what your body can do, how efficiently it can hold on to messages and sort of information and respond to the environment that is throwing that information at it. So, all in all, histones and DNA are like the dynamic duo of life, or as we know it. And I think it's pretty cool. I mean, bacteria, like unicellular organisms, don't have histones. Histones actually are, well, some of the earliest work in histone research was made by David Alice. He won the Nobel Prize for co-proposing the histone tail code with my PI, Dr. Brian Strahl. And that was back in the year 2000. And since then, there have been so many discoveries from that work, and how this tail becomes a toggleable landscape for all these modifications to affect the expression of proteins in your cell and the dynamics of how we live. Anyway, all proteins need to come from somewhere. Proteins are important, and they can be as fundamental as, as life. And so, but the unifying thing about proteins is that they are made in the ribosome. So, what is the ribosome? The ribosome, maybe like vaguely familiar. It's basically what is going to happen in from going from the DNA, from all that histone stuff in the DNA. That is all the library that gets access to be checked out and made into usable, functionable things. And what happens is once it has been printed by RNA polymerase II, the Paul that we were looking at, this guy, yeah, it's like a big complex, but he'll just run and churn. Okay, let's see if we can find a good Paul. Yeah, it's all these different representations of Paul, but they're just bubbles. They're just little guys. Some of them have look at them, just doing a lot. Set D2. Ah, yes. Well, there's a postdoc in my lab who works on set D2. Yeah, really cool stuff. Apparently, it holds the whole nuclear lamin. It helps regulate the on-off switch between making that and breaking that, in a way. In a way that doesn't have to do with its traditional established catalytic role. Catalytic meaning just being one of the enzymes to put one of those marks on the histone tail. Boop. So outside of its boop zone, it has another function to hold up the nucleus from the inside. And so these guys can be diverse. They're they have personality. But looking at how they're made, you have to go from the DNA to be transcribed into RNA. And said RNA will leave the nucleus, the little nuclear basket, and it will, let's say yeah. Okay. So all the DNA is in here, and so you have to let it leave as mRNA. So the DNA will be made into RNA, and that RNA will leave the nuclear pore here. And pretty much as soon as that happens, it then gets captured by the ribosome. So right now it's just a single strand of information. And that information is now the checked out book from the DNA library. And once it gets out of the nucleus, what happens is the ribosomes capture it. Ribosomes are they're cute. They're little sandwiches. So they'll essentially the small guy will find the RNA, and then the large guy will find the small one, and then they'll smush together. And what happens is within this, it becomes a factory. It creates a pocket and a localized zone of order, and from which all these other workers, these transfer RNAs, can sort of buzz along and find the ribosome, and then scan the RNA down here. They can sort of scan it here. Let's see, where's tRNA? These guys, yeah, look at them. They're like little spaceships. So these guys will now find that ribosome, and they'll say, okay, wait, is this right? Is this right? Is this the right code? So there's it's like a little puzzle. And they bring with them, if they get that puzzle right, they bring with them a little amino acid, which is one of the building blocks of life. And there's 20-ish of them. And so, as we were saying, with all these different sort of compositions and abilities and versatilities. So, super cool. Now they're docking, and within the ribosome, there's actually, let's see if we get there's these sites, these little pockets within this little nano factory. And these pockets can facilitate the reaction catalyzing. Yeah, the tRNA also look like these little cross structures. Super interesting. Anyway, they're stitched together here in the ribosome, and then after they'll sort of leave as proteins. And yeah, and all this stuff is happening right here outside of the nucleus in the rough endoplasmic reticulum. They call it the rough ER, the rough ER, because it's bumpy. And there's a smooth ER, and that's where what is a smooth ER? I feel like calcium stuff. I forget what happens in the smooth ER. Protein folding? I feel like protein folding, because what's happening in the rough ER, all these bumpies, these guys are ribosomes. So they're just waiting for messages. And the amount of ribosome is a good estimate of how much total proteins, these workers of the cell, is it able to make? You know, like we have a supply. The demand is coming from in here. The demand is the nucleus, and then here's the supply, is ribosome. So ribosome fluctuations, like in the cell, it is important for metabolism. Because if our proteins need to do a bunch of stuff, they need to kick stuff into high gear, they're going to make a bunch of ribosome so that they can make a bunch of protein to do a bunch of jobs around the cell and remodel and restructure, and they get busy. And that's contingent on how much ribosome you have. So things, if there were within the cell a master signal, some sort of like command switch to say, like a literal toggle of how much ribosome do we want, that'd be pretty cool, because there's a bunch of genes, bunches and bunches of genes, but some of them can regulate and sort of control the expression of a bunch of others. So sometimes there's a very upstream pathway in very important functions. So one of those is actually tied to aging. So it gets its name, the special signal that is in the cell that can sort of make this toggle of metabolism within the cell is called mTOR. So mTOR is another protein. I mean, kind of like the SETD2 or polymerase or histone. It's another protein that does a very specific job. And let's just say mTOR review. Oh gosh, I'm looking at a lot of tabs already. The multifaceted role of mTOR. Yeah. Okay. And oh yeah. It gets its name from rapamycin. And if we were to look at molecular, I feel like I had a molecular review of aging. Yeah, it was here. Okay, cool. Yeah. Okay. Aging is will be a major social problem. Yeah, it's a social problem. Yeah. Okay, so lots of things happen. Let's see. So you get protein balance loss, DNA damage, disabled macroautophagy. So the cell's ability to eat itself and sort of digest components it doesn't need, because you don't want stuff sticking around that is getting in the way of the new function it needs to be providing. So Alzheimer's is thought to be caused by these proteins that have a specific function, which is to shuttle things across the cell from one place to another. Microtubules. Microtubules in movement. So, microtubules, massive little, think of them as buses in the cell. These guys are going like to and fro. Oh, videos, please. Videos, please. Microtubule dynamics. No
In
preview.
this video, we're taking a detailed view on the microtube that is loss of structure, much, and
Little
it will
guys.
becoming
Yeah.
stable.
Okay,
And as a result, it will depolarize rapidly. And this is called a catastrophe. And it will continue until a GTP cap is formed again. And this is known as
Yeah. So they build from one end and then they break on the back end. But in the process, you can get wait, why didn't it just like microtubules? They would have done it way more justice. Oh, I love it. Microtubules. Yeah. Okay. The regulation of microtubule dynamics through the growing tip. Yeah, right. Okay. So microtubules. All right. They do lots of things. Where they go, the cell can go. The cell can remodel and restructure, a large part due to these growing actin tubes. They provide structure for the cell. They can get hitchhikers, but they're like a lifeboat in a big moving sea. And there's a bunch of them. There's tons of them. And it creates order and motor proteins, guys that sort of walk along the cell. These guys, well, okay, that's not in the book. Yeah. They can take chromosomes, they can take organelles, like all bits and pieces from within the cell. They can just take, look at all these devilish shapes. Oh, yeah, okay. Yeah. Neurons, especially, need a bunch of different ways to sort of tangle and stretch and wrap around and rewire. And so it's good to have a lot of directionality to these highways. And so there's a bunch of structural proteins that like kind of guide it and sort of tell it where to go. And yeah, this is what it looks like under a microscope. Little hollow tubes. I once asked the head of the biochemistry department, whether they were responsible or in part responsible for consciousness, as there is someone named Penrose, I don't know his actual name, but Penrose's theory of consciousness is that microtubules and the crystalline structures from within these hollow tubes of tyrosine residues or pyrimidine, I forget what amino acids, allow for computational quantum resonance, not resonance, what was it, like quantum, like super radiance, like some sort of synchronization, I don't know, but he's saying that that happens in cells at like standard conditions or like biotic conditions, and so that other quantum effects can like arise from that ability. I don't know. What do we know? I think it's a little bold to say, but it's cool. It's an interesting, like, thought-provoking thing, because look at these guys. They're doing a lot, and they're super important. Anyway, tau tangles. How does Alzheimer's happen? Because neurons are doing all of this a lot, so how does something go wrong to cause the dysregulation of neurons that are using these to remodel? Where's a review tau and amyloid? Okay. I like spelled everything wrong, but okay. So there are aggregates. So the proteins, instead of doing their specific jobs, so like, look at this. Oh, okay. Yeah. Okay. So don't look at it. This is bad. But it's a thing. Tau is a microtubule associated protein, and so it plays a part in regulating these molecular highways, and so it can get modified just like histone. It can get toggled on and off and on and off with this modification, and that will change its entire like nature. Its charges, its not really its weight. This modification is a really big one compared to everything else we've looked at. It's a really electrostatic or sort of very charged modification. So it occurs on these tau filaments, and the more phosphorylated tau you get, it can form these long stretches. Hyperphosphorylated tau seems to cause these aggregates. Let's see if we can find more aggregates. Aggregate? No. Actually, I think that's what we're looking at up here. Okay. Disturbed cytoskeleton, reactive astrocytes, you get plaques. They build up and eventually become an untangleable ball of mess. They become too hyperphosphorylated, and because of these aggregates, like, it's like a dud, like nothing's really, it's a misfunctional protein, and it does a good job in its early lifespan. So like when we are younger, it does a good job. Like it helps our microtubules go in directions needed for synapse formation and to make our neurons go where they should be going. But as you get older, something about the way that we exist makes these things modify at a higher rate, and some things cause hyperphosphorylation of this, and as a product, gunk in the cell, and then the neurons are really sensitive to that. So yeah, okay, that's a tangent, but we're getting back to growth and aging. Oh yeah, aging. That's where this started. Okay, molecular basis of aging. That's one feature of aging. There are many other features of aging, and they listed here. NAD loss. Mitochondrial dysfunction, disabled autophagy, telomeres attrition. Okay, so what a protein balance loss. All right, so hold on, it's like a little bit cold. I have my deer with me. So protein balance loss and autophagy and mitochondrial dysfunction. That's all cellular metabolism stuff. So people trying to stop aging are using a drug that actually interferes with mTOR. It disables mTOR, essentially. So the signal that can switch the ribosomes on-off, it's a drug that interferes with the mTOR pathway. And mTOR, hold on, I gotta get the molecular review of mTOR. I feel like I totally had it out. Where was it? Oh, wait. Oh my god, wait. That's what I was gonna talk about, though. Perfect. Okay. Yes. Okay. Let me just, like, make sure that this is, like open. Because that is really cool. This is, like, one of the cooler things. Okay. Alright. Aging lysosomes. Yes, absolutely. This was the entire rabbit hole. I recorded a different one, but it was, like, way too long. Because I went on, like, so many tangents. So this is my attempt at, like, not going on as many tangents and, like, being organized with it. Because it will be a breath of, like, hesitant relief to close all of these tabs. Because I will have like communicated it and gotten it like out there. Okay, and also storage space. Okay, yeah, mTOR signaling, by the way. Here we go. Important stuff. Okay, so mTOR signaling networks, mechanistic insights, translational frontiers, and disease. Okay, all right. Pathway. Ooh, okay. It does a lot of things. Mediates mitochondrial lysosome mitochondrial crosstalk. So it's saying, all right, how much lysosome do we have? Okay, how much mitochondria do we have? Okay, how much of this do we have? It's like mediating the things that your body is using to like be alive, to make ATP, the battery of your cell over here, and to degrade things, and to essentially the lysosome is like a trash can, essentially. That's like a huge oversimplification, but it's, for now, we'll just think of it as like a little trash can. So trash can, batteries, like stuff your cells need to be alive. So it's right here in the middle of something. And if we go back up, we can see, wait, another little graphic. What we got? Fermentation, hyperfusion, and so the sort of fusion, the mitochondria isn't like, it doesn't always like look like this. These guys are actually like highly like dynamic and like squishy, and they can fuse because I mean their story is pretty sick. They like us is a we there was kind of a cellular like marriage, endosymbiosis. They cannot live without each other, reproduce without each other, because somewhere along the way they have cohabitated inadvertently or advertently by one party, and then it was commensal, or it didn't really do much in terms of harm for the thing it had absorbed, and so you get these guys called the Asgard archaea. They did a snatcheroo on a bacteria that makes ATP from oxygen when they didn't, because they're like, ooh, you're making lots of bad. Okay, I'll take that. Like, what do you need? I'll invest in you. So like Asgard archaea, endosymbiosis. There's probably a review on endosymbiosis at this point. Dominant contribution of eukaryotogenesis. It'd be cool if there was a T there. The generation of all animals, all plants, fungus, animals, I just got a PDF. Oh boy. Okay. All right. Functionally conserved. I don't see cool pictures. Cool pictures. Okay, so when they're treated with rapamycin, rapamycin reduced the rate of basal apoptosis. Apoptosis is like when the cell is like killing itself in a programmed way, and like the signal has been given, we're gonna shut down now. So the cell's just not doing that as much with rapamycin. So when mTOR essentially is being inhibited just by this drug, it's slowing the rate at which cells are communicating to next cell. Okay. But that's not exactly what I meant, actually. Sorry, guy. Okay, maybe I like asked this question improperly. I think it was the fan, like mechanistic topic of this? I think it was this. Wait, no, this is, oh my god, I found it. Zebrafish paper. Yeah, I would agree with that one. Okay. I remember this paper. Okay. So, what did they do? Zebrafish. Zebrafish are a really good model organism, as in they are the one that has been chosen to get studied, bred in lab. So, zebrafish are now technically super successful. They're the chosen ones that the aliens have abducted and like made like their little test subjects. The reason we've done it for zebrafish is because they have a backbone. It when we're testing a question in molecular biology, we're testing life and like small changes that will affect the entire course of an organism's life. And so, optimally, it would be good for studying like fundamental changes at the level of like a lifespan if we were to look at an organism that like lives and dies quickly. And so a lot of times we'll use flies because they're dispensable. And so you can basically test a big question really fast. Or get like a big screen, a panel. Okay, let's just do a bunch of random stuff to a bunch of flies, and let's look for the interesting like result. And they'll take those flies, and then they'll find what's different from them compared to all the other ones. So you've now found the one like rare mutation that causes a really interesting thing, and so we use zebrafish because they do live like they do have a rapid life cycle, but they're not like unlike flies. Zebrafish have a backbone, which means that their common ancestor, like to us, is a lot closer to like flies, which is like 600-700 million years, like almost a billion years ago. But for these guys, it's less than half a billion years ago. Like before dinosaurs, before really anything was on land. Scorpions may have been on land. Scorpions may have been on land. And dragonflies, not dragonflies. Most things are in the water. We have a common ancestor that had a backbone. The common trait that like unifies all things with a backbone, all vertebrates, is having a notochord, which is like a little stiff part in the back, a postanal tail, a sort of a like mineralized like backbone, as well as like foreign ones. Yeah. But basically, they're really close to us. And that means they share, or are more likely to share, genes and act similarly to us. So we can, when we ask a question and poke at the fish, it tells us more about our own biology and how directly translatable these things are versus something like a fly or an invertebrate, where it's like there could have been a lot of different changes to their physiology as a product of mutations from all that separation that just makes them more different and respond different. So this is closer to getting relevant human information. So cool. What do we do with the fish? So what these guys did? Oh god, the fish. Okay, so he's been poked with liquid nitrogen, or like a cryo. They like froze the tip of this little poker, liquid nitrogen, and then boinked the fish to test for regenerative ability. Because in things with a backbone, zebrafish regenerate really well. Oh wait, another thing about zebrafish, they're a really cool model organism because you can make them transparent. You can make them like entirely see-through into like their adulthood even. We've done that through genetic modifications, but natively they're transparent in their youth, and so you can study using glowing protein. I'll show you, don't worry. Look at that. Oh, that's stunning. It always is. Okay, so this is an overlay of a bunch of different pictures, and this picture is generated from giving the zebrafish using CRISPR to insert a gene information that isn't usually there into into the genome, so that when the protein is made in a specific area, it will glow according to how much of that protein of interest is there. So it like makes these invisible machines visible. In our case, we can do a lot with that. We can do so much with that, and we're gonna see a lot of it. Like, GFP is only the beginning. People have done like insane things to GFP to study really cool things. Yeah, so pretty pictures, zebrafish, they can glow in cool ways. So let's see how we can use that to ask an experimental question. So more different and respond different. So this is closer to getting relevant human information. So cool. What do we do with the fish? So what these guys did? Oh god, the fish. Okay, so he's been poked with liquid nitrogen, or like a cryo. They like froze the tip of this little poker, liquid nitrogen, and then boinked the fish to test for regenerative ability. Because in things with a backbone, zebrafish regenerate really well. Oh wait, another thing about zebrafish, they're a really cool model organism because you can make them transparent. You can make them like entirely see-through into like their adulthood even. We've done that through genetic modifications, but natively they're transparent in their youth. And so you can study using glowing protein. I'll show you, don't worry. Freeze the poker, put the poker on, and then look for regeneration. No injury, post-injury. Okay, they regenerate. They're really good at it. When we think of animals that either like regenerate really fast, axolotls and like salamanders and newts, axolotls are salamanders, but like those guys, the cute squishy ones, that love water and are like they're fish with leg. It's adorable. They have such a regenerative ability. And they've like taken a very winding course to get that way, and for that to have been an available option to them. But it has not in us. We unfortunately, if we were to lose a hand or a phalange, that would be not great for us. We can't. Our body is lazy and it does not want to regrow that thing that was just lost because that's too much work. We have busier things to do, I guess. But let's see if we can like change that. That'd be really cool. It'd be really cool if we could change that. And like we might be able to, is the cool part. But these guys regenerate. We don't know why. But they do. Okay. Actin. Oh my god, it's actin. It's the little highway we saw earlier. Hold on. Oh dear god. Oh dear god. Oh yes. Wait a second. Actin. Actinopurgia. Hi, it's this guy. It's the back. Actin. So they made things glow. They basically took a tissue sample and then applied stains to it so that you can see intensities of different small machines. So they're looking for collagen, which is like hair and whatnot, and actin, which is another type of structural protein. So they're sort of seeing the landscape of the tissues within the zebrafish. So they're getting a different angle of what they saw up here, which is like boink, there's death and damage. So let's zoom in on the molecular level. So, okay, no injury, everything is good and fine. You have actin, you have collagen. Collagen looks like it's sort of supporting in these like interstitial spaces, these like matrices. Okay, so it has a defined structure. Cross sections, cryo-injured. And now, yikes. What goes away? Actin is gone. So cells. It makes sense. I guess there's a lot of cell death, and like the cells that are using actin to make these little highways, there's not highways because there's not cells. I can understand that. So a lot of blue goes away. 4 days afterwards, there's like not a lot of blue, and then 7 days, some blue starts to come back, and then collagen is over here, like as like in a different color, as like a better contrast. All right, and then timeline. Blue is coming back, and there is structure forming with the initial type of collagen that was sort of the foundation. So the collagen sort of looks like it's setting a foundation for the what will become the actin. So like as the cells repopulate, as it's being cleaned up, and then eventually you have like full actin. It's pretty cool. Okay, and this is another view of it. Okay, and DAPI here is it stains nuclei. So anywhere there's cell, so it will be DAPI. And then miles to, I believe that's probably it is green fluorescent protein. So jellyfish protein we've attached to this protein mile 7, and it is an indicator for muscle growth. So anywhere it is like there's muscle. So after the injury, or like you got muscle injury, there's a huge erasure of said muscle, and then it starts to come back. So this intensity of green is a proxy for the muscle regeneration. Okay. All right. MPX. Now, what I'm interested in here is when they used rapamycin. MyoD, all right, SMC, tomato, comparison, expression, generating term. Okay. Why were they doing that? Days post-injury. Zebrafish retain swimming activity after cryo-injury. All right. Interesting. So their ability, mean velocity is the center point. They're like calculating their swim. Oh, that's adorable. Look at that. Oh, that must have been so fun to set up. Distance moved. Okay, so after the injury, they're like, I need a break. I need a break. And then eventually, eventually they're fine. About fine. Their baseline is is 6 centimeters a second. Sorry, it's really cute to me. Yeah, okay. So all these different inhibition of TOR signaling impairs muscle regeneration. One of the common regulators of tissue regeneration is the kinase mTOR. Determine which pathways involved in skeletal muscle regeneration after chronically. We applied an antibody against so all right, this is a player here. Antibody against phosphorylated ribosomal protein, this thing here. This is a proxy. If this is detected, anytime this is detected, it is active mTOR signaling. So mTOR is now going and like talking and doing metabolic work to sort of remodel, get the cell into into go mode to start making more organelles, mitochondria, lysosomes, ready for division, metabolic blue stuff. So this is their proxy for that. So they use an antibody, which is like a small little tag that you can throw at your sample, and it'll stick to only a very specific thing. And in this case, it's that. And then you can apply like a secondary or like another thing that will only bind to the first thing you threw in there. And so you got a hitchhiker and then a thing on the back that glows. And that glowing, you can shoot little laser beams at it, and then the laser beams will emit a fluorescent light, a photon at you, and you can catch it. So you can do that and get an indication for when the cell is going into go mode. So this is a readout for mTOR. And they say that, okay, was so at 4 days, okay, the figure 10 seems important here. Oh, here's where they did it. Oh, okay, wow, okay. I completely missed this. Okay, so this is the pathway. They're like, rapamycin inhibits mTOR, and mTOR is, it looks like mTOR actually activates this. It's still a proxy for whether this thing is active, but it's like downstream of it. So it's a product of, it's evidence that mTOR has been active. So, rapamycin. No, that's good. So this will inhibit its activity. So what do? They test it. This is normal, so everything is true here. This is just regular conditions, no rapamycin involved. Where they a couple different things. So DAPI is nuclei, and then actin. And we know at 4 days post-injury, there's lots of stuff that's dead around that site, around the cryo-injury site. So, but what we do have going on is active phospho, RPS6, and DAPI. And as... interesting. I really would have liked to see more of this, because like this is like establishing like a baseline of how much activation of rapamycin is going on, and they only give at 4 days post cryo injury. And so like, okay, we get that it is active here, and then they're comparing all of that to when they've now applied rapamycin. They're saying that 30 days post injury in control. So this is all up here. This is 30 days after injury. And this group, all of the stuff up here, is with regular, no rapamycin, no drug added, just letting the zebrafish heal. And this is the zebrafish after it has been in a bath of rapamycin. And when they put rapamycin in there, the collagen doesn't properly come back. And it sort of forms... What is that? And the cells, the actin doesn't fully return either. So it inhibits repairing. And the collagen matrix is destructured and very diffuse compared to this organized shapes over here. So the tissue is like structurally changed. It's structurally worse. It structurally has it's been regenerated lazier than just regular conditions, and that is a product of stopping the mTOR pathway from kicking the cells to regenerate. All right, so we have a pretty important pathway, and we have a pretty important drug that interferes with this pathway in a specific way. Cool. I can see why people might think that this could contribute or affect the aging like phenotype. It is probably way more complex than we know, than we understand right now, and it certainly needs, like more testing. But like, I don't know. I get it. mTOR is important. It does lots of things. What is it doing here? Okay, let's see if we can find it right away. Oh yeah, we can. All right, so this is the structure of a lysosome. Oh wait, hold on. Well, actually, we gotta get to this like earlier. We gotta get to this later. Oh gosh, we'll get to that later. Okay. mTOR does a lot of things. Okay, been through that. Yeah, mammals. Yeah. Oh, there it is. Okay. ATF4. This guy. So ATF4 is a transcription factor. So this is the another signal, another protein signal that his job is essentially to blow the whistle if there is stress, and sort of get the protein machinery responding accordingly. And it looks like he is actually mTOR. It looks like he's tied in with mTOR too. mTOR is this master control of even other big players in this. There's a lot of moving parts, but attacking the mTOR pathway with rapamycin seems to be a good way to control cellular metabolism specifically for the process of regeneration. Okay, so now I would really wait, hold on. Okay, now I probably gotta go digging for what I wanted to talk about. So what's another oh yeah, getting back to axolotls. Ooh, aging related diseases. Sorry, I shouldn't laugh at that. I shouldn't laugh, but they made him shake. They made him very shaky, which is pure. That's raw. Yeah, you're right. This fucking sucks. Let's fix that. Histone, epigenetic alterations, mitochondrial, systematic consequences. Yeah, aberrations. Guys, let's stop it. Let's stop it, please. Okay, I'm super zoomed in here. That's fine. Systematic inflammation, yeah. Mitochondrial oxidative stress, metabolism. Yeah. Yeah, so heightened metabolism can cause free radicals. So it's these free little oxygens that will be, okay, let's see if we can see it here. Ooh. Blocked mitochondrial nuclear genome coding channel. Oh, okay. ROS, reactive oxygen species, free radicals is what we'll call them. OXPHOS, damaged proteins, proteotoxicity, release of DAMs. Reactive oxygen species can sort of create disturbances in regular protein interactions, and it just sort of ruins the vibe. And in some cases, it can actually cause. And it's stop that. One of the reasons the mitochondria evolved, some of the essential proteins that the mitochondria uses to be the powerhouse of the cell were actually initially created to basically imprison free radicals in these reactive oxygen species. So they're really good at taking and manipulating oxygen, and proteins have co-opted that to spin. Yay! And now create ATP for the cell and become a little modern. So really cool, unintentional things turn into essential things randomly, given enough time, So getting back to aging and mTOR. Okay, we gotta go digging. I'm basically oh god, the tabs are becoming endless. Mechanistic insights. No. Wait. What is this one? Proliferation and differentiation of osteoblasts. Oh, okay. Well, actually, this is cool. This is also another one. mechanistic. mTOR complex 1 regulates cell proliferation, cell survival, and differentiation in regenerating zebrafish fins. So the other study that did the cryo injury, they're kind of treading the same like ground. Yeah. So they're basically like doing what had been already established in the. So this is cool. It's building off of this literature that came into like a decade earlier. All right, so they're looking for activation of the indicator protein that the other guy used. So anywhere you see green, instead of, I forget what they used last time, but now they're using green. So it's green here, and DAPI is, again, nuclei. So anywhere that there's DAPI, that's a little nucleus. Cool. Okay. And they did more cuts. But this time, it is in, it's still in Zebrafish. I thought this was just an Axolotl paper. Okay, they've made cut. Okie dokie, they made cut. And they let it regrow. And then what should happen, this is their baseline. So they've done nothing except for cut and to see how the internal cells, like how they rewire, and specifically what this green pathway or this green protein's activation looks like in that regeneration process. So what looks to happen is by 24 hours post-ablation, post-injury amputation. What looks to happen is it is broad and is high in intensity, and then it slowly like sinks back down. Okay, interesting. But by 48 hours, there's not a lot of it in the tip. Okay, so it's like hypoactive almost. It's like baseline activated. Interesting, but it seems to like continue for up to 120 hours. Okay, interesting. So it like has a persistent activation. Interesting. Okay, here they're testing different guys. So this is the same mTOR indicator, and then PCNA is essentially it's a protein that will oh goodness gracious. Okay, I'm gonna remember that I was PCNA. This guy's sitting is gonna have to duplicate. And in order to duplicate it, the double helix has to unwind. And it has to unwind. There's so much tension in there. It's bound together. Something has to split it apart, hold it open, and then duplicate both strands as they've been split. So you need a bunch of proteins to make this whole thing happen. You need to like first find a place, and then once that place has been found, all the stuff that's about to like push it open and like get the stuff like docked and loaded on this like railway essentially of DNA. What needs to happen is with this guy, it acts as like the gun essentially. This is like the anvil of like the gun, and so when it's modified here, that's the trigger. And then from that point, it pops the DNA open and then starts the replication process. So PCNA is an indication of if there's a lot of PCNA, that means the cell's like dividing. It's like ooh, we're about to start dividing. Like they're doing stuff. So what they're looking at here is okay, mTOR is going on. This metabolic stuff. Are the cells also dividing? And they see that like after ablation, they're dividing a lot. Yeah, they continue to divide. There's more. So mTOR might go away, but then after 3 days later, there's a lot of division going on. More division and a little bit less mTOR, and it's more concentrated. Interesting. So it might be creating like a pool of. Cool. All right, and then look at that DMSO and rapamycin comparison. So this is similar to that other papers side by side at the end that we saw. I'm not gonna bother trying to find it. So with DMSO, so nothing, no treatment, the zebrafish regenerate fine, and then with treatment of rapamycin after the same amount of time, practically nothing. And so this is a really cool like impact that a single drug can have. Now, does it do the same thing in other animals that can regenerate really, really, really fast? This brother, it does. Yeah, it does. It does do that. I'm like really trying to like. Oh wait, I see the pipeline. Don't worry. Maybe it was here. Oh wait. Okay. You know what? Like we'll use this one. This will actually be quicker. Okay. Um, protein synthesis activation on injury in axolotls, but not in mice. So what they're saying here is like mice don't regenerate as well as axolotls do, which is predictable, but like gotta make a baseline, I guess. So or not I guess, sorry, that sounded passive. They've done good work. What they're looking at here, okay, 0 hours, 24 hours. This is like after they're seeing the state of the area after amputation and what happens predictably in like correspondence with this whole the cells are getting into like high gear and they're starting to divide and they're doing they're starting regeneration. You make more ribosomes because that's exactly what the mTOR pathway is responsible for doing. So mTOR, ribosomes, and predictably after in the area of the cells within like the amputation zone, they got a lot of ribosomes, heavy polysomes. So this polysome, what they're saying here is the complex. They've captured the complex of RNA and these messages, the messenger RNA, as they've left the nucleus and have been smushed together with the ribosomes to make all the proteins that your cell needs. So they're saying they find a lot more of that in the site after activation. So cells are in high gear. Sort of makes sense. So we ODAPI, OpDAPI, cytoskeleton. Okay, so muscle post-activation. Okay, op. What is op? What is op? mTOR. Where do they say op? Where is the part where you explain the acronyms, please? Agrees into op incorporation in all tissues in the amputated limb, including muscles and bone. Why? What is important about op? I don't see op. I could literally use the search bar. Oh, it's here. Okay. To visualize which tissues corresponded to amputation by increasing translation, we injected axolotls with propanolol puromycin. Incorporates into nascent proteins can be detected on tissue sections. Okay, nascent proteins. So this is like a proxy for like how much total like proteins being made. Like in like okay, wait. Moderate. Okay, so this is like they're trying to get a proxy that's quantifiable for like the rate of all the cells they're making a bunch of protein. And so they're using this specific indicator that is in red. So this is in mice. There's kind of a change in okay, but how do they talk about it in the figure? In the mouse, amputations that spare the nail bed can regenerate and rely on the presence of a stem cell niche. Okay, by contrast, axolotl limb does not rely on a dedicated stem cell pool and therefore a distinct regenerative context. Interesting. Yeah, okay. The axolotl does not, yeah, okay. So a significant and highly localized increase in op incorporation a day after the amputation that was restricted on the skin adjacent to the cut site. And it was accompanied by increased proliferation throughout the digit. Wound closure takes up about a week in the mouse, but using op assays, they didn't see any increased translation or proliferation during this process. So think back to the okay. This one? No, not that one. Not that one. The other one. I found it. Okay, I thought I lost it. Yeah. Okay. So they didn't really like reconfigure correctly after mTOR. When they treated the zebrafish with rapamycin, didn't regrow properly because the cells were not creating more ribosome and creating more global protein, so they weren't in this hypermobilized state. But regularly, their mTOR goes wild and it starts kicking things into a regenerative state. So that seems like it might be it's similar to axolotls here. Okay, you know what? I should include this paper of like sort of how this process starts. Okay, so okay, so please verify that I am indeed a human. I request access. I really like this graphic here because he's so stoic. he's like in the limbs make him look like a deity of some sort. He just has like, which is very fitting because I mean they could pretty much almost indefinitely regrow their limbs. Like there's like they're so cool, and not just their limbs, but like their entire like a large portion of their brain and entire organs. Like this isn't just limbs. Like they can regrow so much, and they have backbones, and theoretically they're closely related to us relative to like a fly, an insect. So like there's some secret they've developed, and like they're just better at regenerating. So they're in, we're getting to it. So what they see here is the sympathetic nerves, adrenergic signaling, mTOR signaling. Okay, systematic stem cell activation following amputation follows, promotes faster regeneration. Okay, so instead of at the local area of like what's going on at the site of regeneration, what these guys look at is if there's some sort of signal that is controlling regeneration on the at like a hormonal level, where there is a molecule being released systematically through the bloodstream, and it's like affecting the entire physiology of the animal and is changing whatever cell is like able to be sort of interacted with this molecule into a primed state of some sort. So there's a molecular cue, a signal globally released that can prime the amputation site for regeneration. So what they did is they have two groups. One group, my axolotl chilling. They didn't do anything to him. They waited 7 days, and then they cut his right arm off. Apologies. And then what they did is they looked at how it regrows. Then in the other group, the less fortunate group, I'm sorry guys, this is horrible. Does it like, I'm sorry, like these guys need a statue. They need a statue for what they can do, what we've learned from them. And also their native habitat is Lake Xochimilco in Mexico, and it is polluted. Wait, I'm going to butcher the spelling. Yeah, it has a Z. I didn't expect a Z. I'm sorry. Oh, wow. Mexico. Yeah, his hometown. That's his home stretch. Yeah, it's hydrological basin. The lakes were the home of many Mesozoic Americans, pre-Columbian, Spanish colonial description, remains a lake, urban parks, plant market, UNESCO World Heritage Site. Nice. Oh, that's beautiful. Cool. UNESCO, I love UNESCO. They basically protect things for they're like, this is a special place on Earth. So let's make special efforts to not interfere with the way it is. And that goes for a lot of fossil sites. They're like super important, and they become UNESCO protected. Yay. Anyway, critically endangered species. Yeah, okay. Compromised and reduced state. The accelerating impact of urban. They're listed as critically endangered, but there are many efforts, especially because of, unfortunately, they're like, you're cool for this molecular reason. You're now again an abducted species for our tinkering, and tinker we shall get back to. Okay, basically this is the group that he's the lucky one because he has gotten to keep his arm for 7 days, and then it gets amputated, and then they're just comparing how they are systematically with a couple of questions we'll look at versus a group that has at the beginning he got his left arm cut, and then they wait for a theoretical activation. So if there would be some sort of priming going on, they're trying to catch it in the act. So they've now primed the axolotl in this other group by amputating the left arm. And now they're seeing how he's activated. And so what do they do? What they see is that the group that had been activated earlier, 7 days earlier, regenerates faster. And you can see that at 25 days post-injury. Oh my god, 25 days? Look, there was nothing. And then they have limbs, little digits. What the fuck? Yeah, well, that's really cool. But they say that mTOR signaling had not been established as a part of this regenerative process. So they see that it's working quite well, and that okay, contralateral, lateral. Okay, hold on. EDU. What is this doing? Denervated. They dener. Okay. All right. Oof. Sorry. This one it involves nervous system stuff. Okay. So the idea here is that something in the something about the state that an amputation puts the axolotl in, like the stressed out state, makes for a global, like, or at least in applying to the other arm, faster accelerated regeneration. So okay, poking the axolotl in some way activates it. um, and so activated cells are primed for future regeneration events. Okay, cool. The peripheral nervous system at both the amputation site and responding site is required for systematic activation. Okay. So what does that mean here? So this is the contralateral limb, ODHA. So they're looking in their primed system whether the neurons in there. So we have our like central nervous system, which is like the part of our brain that we are the part of our brain. It's you got a couple different branches of your nervous system. One of them is like, you know, your spinal cord, your brain, your eyes. These sort of like this group of the nervous system that is like executive control and function and like all this sensory integration. But where does the sensory come from and how does it get integrated? You have the other branches that are regulating processes that are not within our awareness most of the time. I mean, sometimes, you know, you feel your stomach grumble and you're like, oh, that is an I am perceiving internal grumbling. But usually all that stuff is thankfully quiet. So that stuff is like the enteric nervous system and the sympathetic nervous system. These other things that are responding to environmental conditions and cues reflexively. So your hair stands on edge, you like sort of maybe turn your nose away at things. They're sort of the watchdogs in all your tissues. So what happens when you use this blocker? So what they did is they injected this drug over here on the right. Let's focus on this panel here. Okay. Can we zoom? Okay. Yeah. So the vehicle in ODHA. So vehicle is essentially just instead of like injecting the drug, they inject nothing. And that is to align the experimental conditions so close with each other that any differences you see is probably going to have to do with the drug, not actually just the process of injecting a needle into the limb. So what they see, what are they looking at here? All right, sorry. I'm going to zoom in real close to this one. Let's download it, and please don't hate me. Please don't hate me, computer. Okay. Perfect. It's probably just better that I have it downloaded. Okay. Vehicle ADHA. Yeah. Okay. So what they're doing here is they're looking at the opposite limb, and then they're looking at the actual like the site of like the regrowing, like contralateral limb. Look at the opposite limb. Okay. What's going on here? Okay, so with this, they're arguing that the activated cells are so EDU here is like it's basically like DNA here, like it essentially stains DNA, or it's a proxy for incorporated DNA into cells. Oh, of course. Okay, EDA biochemistry. I really should have capitalized some things. No, not French. Did you? No, I really, capitalization, EDU, incorporation. It sounds like a company. No, it's not. Guys. I should just put like review paper at the end of it. Okay. Analysis of proliferation, homeostasis. Okay, now the tabs are invisible. A method for fast and sensitive detection of DNA incorporation. So, if there is EDU, like in abundance, if it is being incorporated, and you could quantify those levels, and I think they do that here. Yeah, there you go. Okay. Nuclei. So 14 days after the amputation, when they've injected the axolotls with just like saline or just nothing, there is this level of EDU. So this means there's a lot of regeneration happening because there's a lot of DNA getting taken into the cells in this area, and you can see that here. So in the regenerating area, lots and lots of this EDU like being incorporated. Not so much over here, and this is again a proxy for how many cells are in a dividing state. So you block the sensory neurons, or you kill them essentially. So the peripheral nervous system here, you block it. Not as many things are responding to the amputation. Not as many cells are in this proliferative state. Okay, so the sympathetic nervous system activates peripheral innervation or the responding site or the amputation site, and then surgical denervation via brachial plexus nerve train section. They also like cut the nerve, which I don't really want to look at that actually. I'm like I just have like an image in my head that like I don't really like what. Okay. Anyway, there's familiar names if you have heart issues. Like propranolol. I scribe for cardiology, so I know that propranolol is something that people get prescribed for like their heart rate palpitations. Propranolol, clonidine, Okay, they're doing this to like stop the sympathetic nervous system in this region, because it blocks a channel that is what the nerves use to activate. So they sort of block their communication via propranolol. And what they find is that doing that, the EDU again, they add propranolol, and then like there's less cells in a dividing state. So propranolol stops the regeneration of axolotl limb buds. That's pretty cool. Okay. All right. Where's rapamycin? Where's rap? There we go. Interesting. Okay, what's happening here? Okay, so again, EDU incorporation. So the EDU incorporation is not only affected by by like the nervous system, but also by rapamycin. Okay, so now they're comparing the regeneration rates of the left limbs. Okay, so intact, contralateral, rapamycin. What happens when they? Oh God. Okay. Yeah. Okay. So this is a good like at least. Okay. So up here, this is intact, contralateral. Okay. Intact. So, yeah, okay. Interesting. How many days after injury is this? Oh yeah, okay, this is a solid figure. Okay. Rapamycin does something similar to what we saw in the zebrafish, and it like stops the phalanges from like there's little regeneration after 33 days. That's pretty potent. Yeah, okay. So, okay, the nervous system is responsible for activating the wait, how do they actually draw that conclusion? How do they say that it's like the neurons are activating the mTOR pathway? I forget if I saw that. mTOR signaling is required for systematic activation and priming. Okay, rapamycin. So they just tested rapamycin EDU incorporation. Did they also give it propranolol? And okay, adrenaline vehicle. Okay, so they say that the adrenergic signaling operates upstream of the mTOR signaling in systematic activation. Okay. Okay, so this is another blocker of the sensory neurons, and so what happens is, in the regenerating limb, the blocker prevents the marker that we've been using to study mTOR from occurring, and that is consistent in these blastomas here, at different days post-entry. Yeah, different, well, okay. Yeah, different days post-entry. Okay. Adrenaline. Interesting. Yeah, okay, so the same system, this is the adrenergic system. This is like an adrenaline rush. So they've amputated the axolotl, and it's in a state of like being like adrenergically activated. So the same fight or flight system primes the axolotl for regeneration. So they have a hyperactive, relative to us, adrenergic signaling that facilitates tissue regeneration. Okay. Well, maybe I can't like say that for sure, but like this is this is cool. So we know how these guys are. Wait, hold on. Is this it? All right. So now we are gonna look more specifically at axolotls and what their mTOR is, because this is really cool. But why is their mTOR acting different than the mice mTOR? Why is their mTOR like somehow on a molecular level hyperactive after regeneration? Why is this whole this sympathetic nervous system response occurring to drive their cells into regeneration when it doesn't do the same for ours after an injury? So let's find out. These guys took a closer look at the mTOR of axolotls, of vertebrate animals. So things that are close to us that have this path, this tissue regeneration pathway. Let's look at it. Wait, hold on. I feel like I was just looking at this. Wait, okay. Oh, yeah. Okay, ribosomes. The increased incidence of ribosomes here. Okay. Oh, yay. Okay, so here's a familiar face. It's, but now it's not glowing. Western blots. Okay. Western blots are essentially, each one of these is a different experimental group, a different sample, and it's a different piece of, it's a different sample group. It's a different experimental question. So you basically, in order to get all of this, let's like go on like, okay, hold on. Oh my god, I can totally do this and like have like a new, let's go. Yes, absolutely. Okay. Free tabs. This is dangerous. What was I looking up? Western blot. Okay. All right, so you'll take your sample, and then your sample is super small. It's all the cell stuff. So you're gonna put all of that into these wells, different experimental conditions. So I have one question. This is the cells from an axolotl 0 days after the injury. And now I have another one that's 3 days. Now I have another one that's 4 days, you know? And I will take that, I'll pipette it very carefully into one. I'll label it carefully, the next one, sequentially, and you'll do it in whatever order. And what these big electrodes are doing is now you have this conductive buffer in the middle that will allow you to hook some electrodes up, and you can make a little circuit. And what that's going to do is it's going to throw. So what you've done, you've treated these guys. This is all the proteins and stuff that were in the cells. So now you can ask very specific questions as to like, what the heck was in there? Okay, let me get a good one. Yeah. Okay, it was. Yeah. Okay. So then it basically like it's like a race, but like there's a set amount of power behind the throw, and here's the start line, and they're throwing everything down the body of this gel, or down their lane essentially. So they will run. Okay, start. Wait, hold on. It's really satisfying. Like video. Hold on. Running a gel. Okay. I'll put one in. Yeah, basically, you run this gel or down their lane essentially. So they will run. Okay, start. Wait, hold on. It's really satisfying. Like video. Hold on. Running a gel. Okay. I'll put one in. Yeah, basically, you run all these proteins like in their columns, and the proteins that you're trying to study will appear at a specific size. So all of the proteins in your sample now separate based on their sizes, and so now they're stuck in like a little gel at all the different sizes, and you have a little indicator that tells you where you are size-wise. So the protein that we're looking for in our sample of cells is this thing, the indicator of mTOR. We're like, okay, so let's run the sample, and then let's cut out a little chunk specifically at the size we know our protein of interest to be, and then let's throw antibodies at it that can make it glow relative to how much is in the sample. So the intensities here, all these little bands, they are intense, and that intensity can give you an idea of the abundance of this specific thing in your experimental group. So what we see here is a timeline from 0 hours all the way to 24 at different stages, and they do them in triplicate, I believe. So they're like just seeing like wait, is this triplicate? If it's triplicate, why is it? Interesting. Okay. Interesting. Because, okay, what I'm looking at here is like, you have to like do a sanity check essentially. So the beta actin, you see that 3 times. So this beta actin is essentially a control. So you want to normalize the intensity of your bands to something so that you know that in your experimental question, differences like that you see here are actually relevant and not just a product of there being more total protein in your sample. So you need to equalize it. And so we equalize it by testing and seeing the intensity of a protein that should be in equal amounts in your cells. And then what they say is that after the amputation, what was not there was the activated mTOR. Activated mTOR stuff. I don't know. So, but right after the injury, it spikes. It increases. It starts going, and then it keeps going 24 hours afterwards. So they're saying that indeed there's activation of mTOR. Injury is associated with rapid activation of mTOR signaling in the axolotl. Just using a Western blot to test that. Okay. Activation promotes rapid wound closure. We've kind of been over that. Uridine. Here we go. Here's the interesting stuff. Okay. So in order for mTOR to do its job, it needs to go to a bunch of places. We've seen it hitch a ride with the lysosomes and interact with the mitochondria and regulate physically within the cell these structures that we need to like grow, for our cell to grow and start dividing. So the physical protein interaction of this is important, like how it is doing that. And so what they did, sequence alignment, wonderful. This is pretty cool. So they're looking at the mTOR, like the gene, in a bunch of different animals here. So this is us. This is what our mTOR looks like. So these are just like, this is just like space, because the protein might be bigger and have insertions, and that it certainly does. That is striking. Okay. That is striking. Wow. Wait a second. Whoa, okie dokie. All right. I'm just making a correlation here, but like, list of things that cannot regenerate much at all. Human, sheep, mouse, chicken, dog, electric eel. Zebrafish. Zebrafish can regenerate. They're pretty good at it. But they don't have anything unique in these spaces, is what we're looking at. For now, let's just look at these empty spaces, because in geckos, toads, clawed toads, caecilians, it's a type of amphibian. It's like the branch of amphibian, the third secret branch of amphibians that is like, oh, they're so strange. I'm gonna look them up because they're super strange and super cool. Okay. I'm just gonna look up what their skulls look like. Okay, it's additive. Hold on. If you were to see one of these guys in the wild, they might really just look like a worm. But they're like, oh my god, they're so intense. They look like dragon skulls. But sometimes they'll have like ossified eye sockets because they are fossorial, as in like they're into ground that. They be squirming and worming, and a worm-shaped thing eats other worm-shaped thing. So these guys have a steady diet of worm. Gnarly. Absolutely insane. But their skulls are like terrifying. There's nothing worm about that. There's nothing worm about that. That is like the thing from SpongeBob, the Alaskan. Is it the Alaskan? Yes, I think it is the Alaskan bullworm from. It's a song. I don't think it's that. Is it that? Yeah, it is that. It's this guy. Charismatic. This isn't accurate though, because they actually have shovel tongues that they use to. I'm moving on from axolotl or from those things actually, because that's where the fun of Sicilians like kind of stops. Our definition of fun. Axolotls. Okay. Interesting. They have a little blip. So these are all amino acids. They've been stitched together in not only like a different way. Well, actually, hold on, wait. Okay, so all of this is the same. Whoa. Okay, so if you're looking at these things, sometimes there's gonna be areas that like they stay. Like this right here. Sorry, if you look like the far end right here, that has stayed for some reason. Like it has not changed through a lot of evolutionary time, from humans to geckos to fish. That's stuck around. So maybe it's dangerous if you play with that part. If mutations occur there, that might get weeded out of the population real fast because it's real bad for some reason. So everyone's behaving, except for now, innovation. There's 2 inserts in axolotl mTOR. And it looks like it's common. This insertion is common to newts and fire salamanders. Interesting. Whoa, okay. Is it different at all? Has it changed at all? No. Okay, so this entire thing here may have been an ancestral like insertion. Maybe a virus inserted itself, or well, axolotls have gone through salamanders in general have gone through a wide variety of duplication events to their entire genome. So when you duplicate a gene like more than it should be, sometimes that's really harmless. Having an extra copy can be really helpful, and it can be really helpful sometimes because now you have a copy to play with. So the other one can do its regular job, but now that you have a copy, it can just acquire mutations. So even if inserting something here would be bad, evolution now has time and a sandbox to play with this duplicate of the original one. And what might happen is that it'll figure something out after enough time. Something will happen to that duplicate, and it will maybe become the main thing, or it'll maybe become something that has a new function entirely, or helps the other one do its function better. So who knows what this insertion of building blocks is doing in these 2 regions. So they go on to find out. And what they find is that, okay, Okie dokie. Oh yeah, okay. There was an important thing here in the supplemental. Oh yeah. Oh yeah. Oh, this is exactly what I was like looking for. Oh, this is the fucking insane part. Okay. All right. Yeah, here's the kind of mind-blowing part. In order for mTOR to like what they did is they essentially created a human axolotl mTOR hybrid. So they stitched the things together using CRISPR. They inserted it into this HEC293T. Anything you see here, all of that is that's human kidney cells. So this is all now in human cells, not axolotls, but it's using human cells that have been modified to express axolotl mTOR. Okay, what happens? What differences do they note? Okay, so here's regular, not modified kidney cells. What happens is mTOR, it has a steady state. Okay, wild type, no control, lysosome tracker. All right, lysosome tree model. Okay, where is mTOR? Where is it going? Okay, merged happy. Okay, the lysosomes are acting a lot different. Axolotl insertions promote the lysosomal retention of mTOR. Okay. So mTOR, in order for it to do its job, needs to go to the lysosomes. So lysosomes are, let's sort of get like a small refresher. Lysosomes are these structures within the cell that are kept at a lower pH, so they're a bit more acidic. They have they have ions in them. They have enzymes in them that can cut and sort of act as the trash can. They can sort of degrade things that need to be degraded. And so in the process of like growth and sort of getting rid of old things to get rid or make for new things, this is important. And so as a product, like how it works is mTOR. So mTOR is physically here with this big protein here, this big protein complex. It's physically here interacting to kick the cell into a regenerative state. Okay, mTOR. And there was, I feel like I had another interesting like thing about that. I feel like it certainly was tangential, but like, okay. So I just want to see like what usually it's doing. Deptor, pick, raptor, the bridge horn. This like, it just becomes some sort of alien topography, you know? It's, thank God we can, and rapamycin, here we go. He's getting in the way of this interaction as a part of its catalytic site. So mTOR will do something and it will catalyze something, and rapamycin is just pew, right in the way of that. So cool. That's a mechanism. All right, bigger complexes. What is it doing here? This is a lot of information. What are we doing here? In response to amino acids. Okay. So it can respond to a lot of things to regulate the gator mutations. Okay, these are all pathologies associated with mTOR. I'd like to see it in the lysosome. Okay, there's a lot of talk about the nucleus here. Okay, so these things are anchored to the lysosome by this regulator complex. Oh, that's adorable. This is a name. Late endosomal lysosomal adapter, mTOR activator. Okay, so point is, in order to like metabolically like sense the state of the cell, mTOR is physically tethering a couple of other factors at the lysosome. So it is docked at the lysosome. And so that is in the process of it being activated. Hold on. All right, let's get back to this paper. Okay, so they promote a portion of the lysosome being retained, or sorry, a portion of mTOR now being preferentially retained at the lysosome. Okay, yay, here's where they do actual characterization. All right, so now they're testing the chimeric human kidney cells that have axolotl mTOR. And they're saying, describe, okay, cells were traced, traces of growth media and starved. Okay, they starved the guys. Then they diluted the media. Okay, stimulation solution. Okay, so they made them starve. They put them in stressful conditions. So they're assessing the capacity of mTOR, the chimeric mTOR, axolotl mTOR in human cells to sense nutrient starvation and stimulation over time. We seeded cells as above and then incubated. Okay, rapamycin. Oh wait, am I in methodology? Am I in methodology? I might be in methodology, and I'm not seeing. I want to see. Yeah, I'm in methodology. Oh yeah, I'm deep in the methodology. Okay, sorry. I'd like actually. I mean, that was actually cool. I should probably go back there, cause like interesting. But what they're saying is okay. Better able to discern differences in duration of exposure to nutrients by modulating the mTOR's activating like its off-on switches. So axolotl mTOR has the remarkable ability on amino acid titration to not exceed the upper limit of activation of the mTOR. So it's distinct and not functioning in the same capacity as wild type or an oncogenic mammalian mTOR, while possessing extraordinary resistance to oncogenic transformation. So they don't get cancer because their metabolic pathway is entirely different from ours. Wow. Okay. All right. Yeah. Uh-huh. Okay. All right. So when mTOR is like doing all of its things, it forms a dimer, as in it's like stuck together. It's like smushed. It's in a complex, and that complex forming is like a step in the way. And if you can get if you can preferentially be in a conformation that is close to the reaction you're trying to facilitate, then it takes less energy for that thing to occur. So if you want to sense nutrients faster, being pre-assembled at the place you're supposed to do that job is going to help out a lot. So what they found is localization. Okay. So they observed that axolotl mTOR binds significantly more Reb protein at the lysosome than regular human mTOR, suggesting that it preferentially exists as a fully assembled mTOR, which they say. Then this would account for the hypersensitivity to nutrients and constitutive localization to the lysosomes, as both of these rely on the fully assembled mTOR complex. That's awesome. Okay, trade-off in regeneration. It's possible that these changes is key to the metabolic relay may have affected evolution and adaptations of amphibians. So maybe this was evolved as a need to axolotls are feisty. I mean, they're aggressive. Like I've seen them eat worms and things, and they are they're adamant on what they're eating. And so if they just so happen to adamantly eat, oh, and Lake Tsuchimilko, if the conditions are poor for a long period of time, you are going to be fighting your neighbor for food for a long period of time. And so that means, let's see, intraspecific, let's see if I can find, like a word search. Okay, powerful, this is, okay, nutrient sensing, yeah. Yeah, well, look at that. These findings further suggest the possibility that engineering an axolotl like mTOR may allow us to promote wound healing and perhaps even tissue regeneration in mammals without triggering oncogenic formation. So please, no cancer, but let's regrow with the attitude of cancer. Please, let's prevent ourselves from going rogue. Let's just give them like axolotl superpowers, because they have a hypersensitive mTOR. That's cool. I'll look at more about that later. Okay. So now we have the sequence. We know where the superpowers come from, and we know the nature of said thing. It looks like that. It uses these building blocks. Now, what I would really like to do is transfer that to a human up here, because we don't. Unfortunately, we don't have that. I think actually, it's really it's kind of adorable how they phrase this. It's like evolution patting us on the head. Invertebrate. Please wait. Okay. Wait. Okay. Okay. The insertion is otherwise absent across metazoans, and the mammalian sequence appears to be ancestral, as it is also found in invertebrate species. So we are stuck in our mTOR is stuck in the Stone Ages, compared to axolotls. They've taken it and ran with it. So we should be thankful. Yeah. Well, wait, hold on. No, we should not be thankful. This is mildly concern. It's not concerning. It's just because we wouldn't know. We do great things without regenerative capacity of our limbs. Fine, whatever. Take your wins, axolotls. Yeah, actually, they're not taking their wins. The reason that they have this is probably because the pollution in Lake Tsuchimilko is creating a selective need for them to be able to regenerate, because their arms are being taken off a lot. And having all your limbs is really helpful for finding a mate, locomoting, finding food, doing all the things. So probably like a tragic backstory to why they have these insertions and are able to like intensely sense nutrients and put their body in a hypersensitive state of regeneration at both the nervous system level and the molecular level. But we can harness that. We know the nature of this difference, and so there's a really cool paper. Where is it? Did I have it out here? Was it out here? Tab. Very predict. No, no, not that one. Not horse evolution. No, I'm stuck. Okay, this is like purgatory. Okay, what? We're almost there. We're almost done. Organ conformal kirigami device for gene therapy. Okay, all right. The pocket. Let's put a thing in a pocket. Okay. Now this is a really cool paper. It just came out. Get access to my institution. I know I have access, and now we all have access. Indeed. All right. Download this PDF. Oh, no, I just got fushigi'd. All right. Cool ass paper. All right. An organ conformal kirigami structured bioelectric packs for precise intracellular delivery. All right. Tabs are invisible. Good sign. We're progressing. All right. So, what they did is they made a thing. They made an innovation device. There it is. It goes over an organ like a sheath, a coverage, and in this case, an ovary. And they're sort of highlighting what this all does. Has a couple different layers. PDMS. This is essentially the electric like control panel, like the system that will activate the delivery and is responsive, wirelessly powered. Cool. This is all good stuff because you can put it on an organ and just boop boop. Like theoretically, I don't know how you replenish the stuff. I don't know, like if you were trying to deliver, like, I don't know, like how much can you store in this? I don't know. It's probably here, but for now, what I would like to talk about is no safety concerns. Yeah, you're right. This is true. From vaccines to innovations. Okay, I think this is a review. No, gatekeeping, please, my weakness. Review. But this is my, yeah, look at that. You just type in review and then it'll give you, okay, lipid nanoparticles for messenger RNA delivery. So we talked about the messages that the cell uses to make these proteins in the ribosome. Let's just give some messages. Let's just pass a message along to a cell, tell it to make something, and then make it. Let's do that. Is this feasible? Is this like science fiction? These are all the different constructions. Lipid nanoparticles, these guys will assemble with each other in a way that will make like a circle. They have a part outside that is like, it loves the water, and a part inside that doesn't. And so all the tails will stick together. Like, oh my God, guys, I hate the water. I know, right? And then like the heads are up there, like trying to like go, like they're associating broadly. And so they make a spherical shape. And in the process of that occurring, we can put messages into the lipid nanoparticles. And have we done that? Yeah, actually. The COVID vaccine. So the COVID vaccine essentially works. I think they have an article on it. Yeah. All right. So this is the like messenger RNA, the message that the virus would be using. So this is the virus here. And this is where it will attach to a cell and will like insert itself, and then it will go into the genome and start dividing. It'll try to use your own cells for its own replication of all of this material, and that is stored within here. So what we can do is we can take not even like the DNA of the virus, but just the message that codes for only this outside protein. Because if our cells know what the virus looks like, then we can take care of it and fight it. So what we're doing is we just used the message that codes for the spike, and then we've given it as a vaccine within these lipid nanoparticles that we can make, if injected it, and our cells now have the messages our immune cells now have the messages to make weapons against the COVID vaccine or the COVID virus. So cool. We can use lipid nanoparticles to deliver messages. So when we look at going back to the pocket, we can use lipid nanoparticles that we can put in these things, like these little plasmids, where like we can put messages. I don't even know if they need nanoparticles, but like you can put messages in this layer and then deliver them to cells of interest with a remote control. And let's just initially see how they do that. This, like, the device is so cool, like, as it is. They're like, this is the competition. Oops. Get them out of here. And it's, wait, hold on. This is fun. Okay, as far as I know, it is working. And we are now recording the screen again to finish. Oh, wait, okay, hold on. Okay, and we are now back. I breached my 2-hour self-contract, but I will weeball, and I think it has actually allowed there's cool stuff. I'll make little chapters, I think, so it'll be a lot easier. Anyway, non-conformal contact. So they're like, this is the competition. What we have going on here is this sort of tessellated surface here, kirigami tessellation, so that it's able to morph. And this is a stress and strain curve, radial strain. So how much like strain is the material experiencing? Intensity. I can't do strain. I wish I knew strain curve. Actually, quantitative conformality analysis. I see. Like all the corners start to touch when it's being like stress is being like a stretch is being applied in any direction. We pull partially conformal, so it's very adaptable because these squares. Okay. Yeah, like it's seamless. That's uniaxial step stretch. I see. Wow. Failure strain, theoretical. Okay, so this is all the engineering, experimental sort of analysis of the structure all fit together. Okay. Wow. Portion, ovary. This is just how it fits. Confer high efficiency and uniformity in pocket. Wow, okay. Rabbit eye. Oh my god, they covered the whole eye? I mean, what? Okay. Oh, alright. Yeah. What is this? What is going on? Wait, sorry. What? How are these images being taken? What is this? Pocket intensity. Host cells. Yeah, okay, so you can fly this thing through a bunch of different structures. Okay, so now they're going to test gene delivery. So whether you can efficiently get messages to these cells that previously didn't have them. So, okay, this is like they gave a rat GFP, and so they're testing. This is another Western blot. This is loading control. So all these should be equal across the different cell types or their different conditions. And this is the question they're asking. So these are like they put the pocket around the ovary and they essentially delivered it. So they remote control delivered the DNA plasmid that is now inserted into the cells of the ovary as making the protein. And so they're checking to see if that protein has been made. And what they find is that in the ovary, GFP is certainly there, and they're measuring an intensity here with this sort of heat map, or in like I see it over there. Wow, that is so cool. Okay, and then here they're checking GFP intensity in the ovary, tissue surface, tissue section, pocket. Wow. So the yellow is the overlap between the green and red. So where the pocket is activating, is expressing, is actually has inserted the messages and is making those cells like express both GFP and mCherry. mCherry is another fluorescent protein. Interesting. That's so cool. So it's saying that where there's GFP, there's also overlap of mCherry. Okay, so that's a product of the delivery here. controllable and efficient in vivo gene transfection by pocket, and the relayed delivery strategy via EV. EV? Endogenous virus? What are we talking about here? What does EV stand for? What are you guys talking about? Guys? Am I silly? What is EV? Oh, these are like just the particles. Okay, endogenous vesicles. What actually is... Okay, so I'm assuming these endogenous vesicles are similar to the lipid nanoparticles that we were looking at earlier, and they contain all of the cargo that the cells would receive here. P-derived messenger RNAs. So they're getting electroporated. So there's like a shock that goes on to temporarily open some of the doors that a cell might use to gate everything else usually. So you're shocking it, so you're making a little a I've reached this word, like a disturbance in the membrane. And so that's enough space for these plasmids to fall in and start actually making the GFP that we see them to do. So once this device is on, GFP happens as a product. We can control that. Okay. Okie dokie. All right, so here's what they did now. So BRCA1 is a protein that has been widely studied. So you see it here, and you see it here. You see it a lot of places. BRCA1 is a tumor suppressor gene. Is what category of protein it would fall into. This guy is super important in actually fixing double-strand breaks. So double strand breaks are the worst kind of like DNA damage. So let's see, BRCA1 for DSP repair. Okay, so this is another protein that like I think elephants have a unique version, kind of like how we're looking at like axolotl. Oh wait, is this, who wrote this? Did Ramsden write this? No. Okay. Okay. New twist. Now I'm gonna look out because I know a guy in my department who like, this is his stuff. Rocco wanted to. I pestered him with a bunch of questions one time. Well, multiple times. He was the head of a course that I was like, my favorite course actually. It was really cool. And taught a lot about this stuff. Okay, so BRCA1 is here. So okay, what needs to happen? You got your DNA, it's all linked together now, cool. But something like gamma radiation has now like a sort of like a bullet with such high energy has like smashed the DNA, and now it's just two strands. So these guys, this is horrible for the cell. I mean, you can't like, if you try to like make anything from that, repairing and like filling in that gap can do it improperly. And so like sometimes like the fixing process, not good. But BRCA1, it has its own fixing process pathway like this all, and it's very efficient. It's very good for the maintenance of our healthy non-cancerous cells, because if cells start to acquire mutations, that could be in a really important spot that could cause cancer. And one of those spots would be in the gene for BRCA1, the thing that makes the guardian, the thing that makes him. And so if you don't have that, your cell is gonna acquire a bunch of double strand breaks, and that's not good. Yeah, so it's needed for the crossover of like the two different strands to sort of make this mingling repair. So you need BRCA1. BRCA1's it's good to have BRCA1. And so what they do here is this is just the control. They haven't really done anything. Another Western blot to see that protein levels of actin are really consistent across all the groups. This is in all ovary cells. This is ovary cells of the two different groups of mice, one that doesn't have the pocket and the one that does. And they indeed have tested it. The only place that BRCA1 has appeared is in their group. So cool. And this is gamma H2AX. When the DNA here, this DNA is DNA plus histone. So when the DNA has been cut, histones get modified, phosphorylated. We've kind of talked a little bit about. It gets modified, and that becomes a beacon. That modification spreads like wildfire on the strands, so that now these strands are identifiable to the cell for, oh god, we gotta repair that. So they're testing for levels of that in these different cell types. So what they find is there's, generally speaking, more double strand breaks. They're using gamma H2AX, the modified histone wildfire, as a proxy for how many double stranded breaks are going on. And it looks like lots when there's no BRCA1 expected, and then a little bit less when there has been newly administered BRCA1. Very cool. Okay, so comet tail DNA, looking for damage of the DNA. Okay. Okay, they're staining here the tissues. Okay, this is an important one. Okay, so what they're saying is that after 150 days, how many of the how many of these mice develop tumors just as a product of existing in a world without a tumor suppressing gene? And eventually, all of them, there's 0% that are tumor free. All of them develop some form of tumor. However, the ones that have been administered this pocket never dip. They never develop a tumor. Now, they could have extended this for longer, but I mean, how long do mice live? No, they could have extended this for longer. I'd like to see that. That would be cool. But and then they tested for doses. But this is like super exciting because like they now have prevented, like if you were genetically susceptible to like humans, like BRCA1, like not having an allele, a copy of BRCA1 is a major like risk factor in like human existence. For like if you do not have one, it can be inferred that you're at a higher risk for cancer, certain types of cancers, because your cells are not able to fix their DNA as properly, and that can lead to the cell going rogue because of misinformation in the library. It has been disrupted properly. So now we've given an organ back, BRCA1. That's amazing. That's like genuinely incredible. Like that's so cool. Oh my god. Yeah. Okay. What are they testing here? Improved ovarian function. So now they're like seeing, okay, what's actually going on in the organ that has BRCA1. We know it's not getting cancerous, but what's going on there? And it looks like it improves their function, and they measure that by looking at what? Estrogen. Okay. How well they're able to produce the natural hormones they do. Okay. Okay. Non-coding. Look at that. Yeah. So estrogen levels return to like the wild type, so regular mice. Wow, that's awesome. That's incredible. Okay. So here we have the clinical feasibility of a device that can be used to deliver messages of our design to an organ of our interest. And say that organ of interest is in dire need of more cells and is having difficulty regenerating because of our Dark Ages mTOR that has not evolved these insertions at the, oh dear God, oh dear God, where's the, okay, I think it's this one. Yeah, it has not evolved these insertions. We can use our pocket device to deliver axolotl mTOR. The only thing, all we need to do is get axolotl mTOR as a little message to our cell of interest, or our organ of interest, and now we can potentially put our human cells in a state of hyper-regeneration. And yeah, that would be pretty sick. Kickstarting our ribosomal biogenesis, making all the right mobilizations that we've seen before start happening in human cells that wouldn't ordinarily do that. So yeah, that would be really cool, and like it's all thanks to like modern technology and axolotls. I mean, like, hold on, like these guys, like the real deities here. Look at that. Yeah. So theoretically, axolotls can help us regenerate at the level of the cell. Now, this is putting axolotl mTOR in a living animal has not been done. It's only been done in that in those kidney cells that they used, but those are human kidney cells that showed signs at the level of the cell of being in a heightened state of metabolic sensing and regeneration. So this would need to be done in mice, in zebrafish to see if having these insertions can confer the same levels of regeneration. Probably mice would be better rather than zebrafish because they already regenerate fast. So do this in mice. See if they regrow faster. Inhibit with rapamycin, same way. Confirm that it's as they've been doing. And then, well, I don't know, the whole clinical like feasibility and pipeline, the Pocket people are already working on this. But theoretically, you might be able to transfer a little bit of superpowers from these guys, and that would be really cool. So yeah, I mean, this is pretty cool. I mean, someone's gonna eventually do this, so just a matter of time. So there's hope for people who are missing a limb or have some. This could be applied for like neurodegenerative diseases as well, potentially. Actually, I mean might mean you have a pocket on your brain. Probably not a pocket. Probably like who knows. There's if we can administer using one of many of these like delivery technologies out there, Axolotl mTOR to tissues that may need regeneration, we might actually be able to help them out. Modern technology and biotechnology. So yeah, thank you guys for joining me on this rabbit hole. Yeah, this is cool. I don't really know how I'm gonna do like all these videos, but like this is fun. And it's nice to see different labs and different stories coming to say some of the same things. So rapamycin working on this particular pathway has been validated in a bunch of these papers that we've looked at. And because that science is repeated, we can now run further with new questions. And that is always exciting. So hopefully I'd like to continue this sort of trend and see all like how accessible this stuff is, like, and how exciting we should feel about the future of science, because that's pretty important, is maintaining despite everything, science is always going to be exciting, you know, and everyone has good ideas, you know. There's a lot of fun stuff to find out here, and like there's a lot of silly graphics, like there's silly graphics, and then quite yeah, and then they're regenerating, so it's like less silly but like adorable. He's really derpy, man. He's like incredibly derpy. Yeah. Anyway, see you next time.